Welcome to Click for IB Biology. We're in the topic of chemical signaling, signal transduction, and in this particular video we're going to be looking at localized signal transduction, in which the signal only passes over a short distance. In fact, in this particular case we're going to be looking at neuromuscular junction distances of 20 to 40 nanometers. And the diffusion of the ligand acetylcholine. There's a blank version of this presentation, a PDF. That's linked in down below in the description. There you'll also find 10 questions with answers, and you can use those to check your understanding of this content of the syllabus. So let's look at localized signal transduction. So the particular location that we're concerned with, this is a motor nerve or a motor neuron, and there's an impulse traveling down the axon in this direction here. And the nature of that impulse is actually a wave of sodium ions. These arrive at the ends of the nerve cell here and form a junction with the muscle fiber here, and we call that a neuromuscular junction. And as I've previously mentioned, the gap distance is 20 to 40 nanometers. And in this particular case, the ligand is a chemical called acetylcholine, which is stored in the ends of the nerve cell in vesicles as shown here. Now these vesicles will migrate to the end of the nerve cell membrane, triggered initially by sodium ions. So these will flow down into here, and through a triggering mechanism, these will cause the vesicles to migrate to the cell membrane. Higher level candidates should be aware that this topic is dealt with in more detail in the next topic, which is neural signaling, and also in muscle contraction. So the information here should be combined with that to give you a more detailed knowledge than the simple core of the syllabus. So back to our story. In the motor nerve, the nerve impulse, a wave of sodium ions, arrives at the end of the nerve known as the synapse. The vesicles containing the neurotransmitter substance, the chemical which is going to act as the ligand, is contained within the vesicles, and this is called acetylcholine. The vesicles fuse with the membrane, and the acetylcholine enters this space here, which is called the synaptic cleft. This is a space between the nerves, the motor cell, the nerve cell, and the muscle cell, which is here. The distance, as previously explained, is very, very short, 20 to 40 nanometers, and therefore the diffusion speed is almost instantaneous. On the postsynaptic membrane, that is the muscle cell membrane, there are receptor molecules known as nAChR. So this actually stands for Nicotinic acetylcholine receptor. So we can abbreviate that to nAChR, and they will be in a closed state. So this is a channel protein, but it's actually closed. And on the surface of this channel protein, there are receptor sites for acetylcholine. So the acetylcholine diffuses across the gap here, and the acetylcholine will come in and locate onto the two receptor sites on the transmembrane protein. And this will cause a change in the conformational shape of the protein. The protein will change shape, and the result is that the channel through the protein opens up. The consequence of this is that sodium ions accumulated on the outside in the synaptic cleft will rush into the muscle cell, like so. Now this will therefore cause what's called a depolarization of the muscle cell, and the wave of sodium ions will accumulate until it reaches threshold levels of approximately minus 55 millivolts. And within the muscle cell, this will then lead to the release of calcium ions from the sarcoplasmic reticulum. So if one studies the muscle structure and muscle contraction, we find out about sarcoplasmic reticulum storing calcium ions. This is the muscle equivalent to the endoplasmic reticulum. We call it sarcoplasmic reticulum. And with the arrival of the sodium ions there, the calcium ions are then released, and they become a secondary messenger molecule leading to the contraction of the muscle. And so candidates will wish to link the detail that I've just given through to both neural signaling in the next topic and to muscle contraction. So this is an example of where the ligands are operating over an incredibly short distance in a very localized manner like so. We'll now go on to look at more distant signaling, chemical signaling, and signal transduction in the case of hormones. There's a chance to subscribe to the channel, always appreciated, folks. And there's the playlist for your convenience so that you can see all of the videos in this particular section called chemical signaling for higher level students only.